So the Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in the webinar series. Please see the website for more details. My name is Vicky Higman. I'm a structural biologist working at the University of Leicester, and I'm part of CCPN, the Collaborative Computational Project for NMR. I also chair the Biochemical Society's Research Area Panel on Molecular Structure and Function, and it's in this capacity that I'm chairing today's webinar. We will today hear from Dr. Björn Burmann, who is Associate Professor in Chemistry, oriented towards life science at the University of Gothenburg in Sweden. Björn's group investigates macromolecular protein machines underlying essential cellular functions using high-resolution NMR. They aim to elucidate their function at the atomic level in order to understand their dysfunction, several underlying neurodegenerative diseases, and also cancer types. Björn has won several prestigious prizes for his work, including being selected as an EMBO Young Investigator in 2020, and has been recognized for his pioneering contributions to the determination of structure and dynamics of chaperone client complexes at atomic resolution by solution NMR. So it's a real delight to have you with us today, Björn, and I'm really looking forward to hearing what you have to say about your work.
Yeah, thank you very much, Vicky. I hope my slides are nicely visible, and thanks a lot for the kind introduction and the opportunity to speak of the work we are doing here down in Gothenburg. So in my what we are interested in my team is that we want to understand how proteins actually function on the atomic level. So bringing it a step further than just a structural investigation. And for this, we're using NMR spectroscopy on kind of complex and difficult proteins. And kind of in general, what we are doing in my lab is kind of different aspects. On the one hand, and this I will be talking to about today mostly, is on the left-hand side that we are looking at different complexes involved in transcription-associated complexes, lately related to DNA repair mostly, as well as kind of protein quality control, in particular also in mitochondria. On the other hand, kind of we are aiming to bring this also further just from studying just the isolated proteins, more in the cellular context. We also do a little bit of in-cell NMR approaches, which were kind of pioneered by Phil Zelenko to a certain extent and Volker Dörtsch in Germany over the years, and we try to also implement them to our studies. And some things we are also trying to develop as kind of in situ approaches to get a little bit resolution. But as these projects are in the progress, I will not be talking so much about these things, but I'm happy to discuss options and things. But I will rather focus on our work on understanding different cellular machineries and how they're regulated and interacting with each other. So the first topic I would like to touch on is actually a bacterial system, and kind of the very important question, which also relates to kind of us as humans. How are actually cells cope with kind of damage to the DNA or so-called DNA lesions? Well, this is a very important kind of question, and this was also awarded in 2015 with the Nobel Prize. And in particular, if you look now on the left-hand side, kind of this model system with the most famous MFD protein was kind of investigated by Ashish Sankar, who was one of the three kind of Nobel Prize winners in this year. But over the years, it became clear there seem to be many different ways how cells can cope with this. They are understood on the genetic and biochemical level, but there also seem to be several kind of interconnecting things that certain transcription factors, like here listed in the center, the NAS-A protein, might play a prominent role in different aspects. And having a background during my PhD and kind of studying bacterial transcription regulation, I became interested in this, and we started to work on several of these proteins. They have one common thing: these kind of regulator or proteins which facilitate the repair are actually large on the NMR side. There are at least 40 kilodalton to up to 70 kilodalton, which makes them kind of a bit of challenging, and you have to play a couple of tricks, which I also will introduce to you how we can study these more complex systems. Because if you still open your classical textbook, the standard NMR protein approaches are kind of feasible up to 15-25 kilodalton, but beyond you have to play some tricks and think about kind of the systems you choose. And kind of the first protein which we kind of tackled in this complex was a small DNA polymerase, a so-called DNA polymerase 4, called DIN-B, which is a so-called translesion DNA polymerase, which kind of can cope with kind of damage in the DNA and kind of fill in gaps. And in particular, it became clear about 10 years ago, it's not only associated with replication, as it was initially thought, but also transcription, where it in the non-template strand can fill in gaps that RNA polymerase can continue. And we became interested in studying this protein. So what you normally do as an NMR spectroscope is to initially kind of express your protein and look at the spectra. And this is kind of mainly the work of Damasus, the former PhD student in my lab, together with another PhD student, Jens, who did a lot of the analysis. So, and despite the size of about 40 kilodalton, we got kind of these, let's see, just get the laser pointer. So, and we got this well-dispersed spectrum, which tells us every kind of these kind of dots represents a amide in the protein backbone, besides for every amino acid besides proline. So what you can directly see, we see kind of the expected number of peaks, which is great that we can see the whole protein, and there are not clustering in the middle or anything which would be indicating large unfolded species, but they're nicely well dispersed, indicate a well-folded protein. Well, despite the fact that we got decent spectra, kind of the assignment, kind of putting an amino acid to each of these dots, turned out a bit more tricky than we expected. So we kind of made a lot of subconstructs, kind of exploiting this modular approach of this protein, and assigned all of them, which we could kind of express and purify, and which were stable. And we could then transfer all the assignments kind of back to the full-length protein. And this kind of matched well. The plot here at the bottom indicates kind of helical and beta sheet segments, which we can kind of extract out of our kind of the chemical shifts of the side chain C alpha and C beta atoms. And this matched well with kind of what we observed in the static crystal structure. So this was our starting point. We now know what each of these resonances of these peaks belong to. Now we can probe kind of them and see how they change in terms of interaction and dynamical properties. And dynamical properties is, I think, what kind of NMR spectroscopy on proteins is actually, to a certain extent, the best method you can choose from. Of course, you can also study dynamics with several other techniques. But on the other hand, NMR gives you such a rich kind of toolbox, which we just had here in a small review, just looked at different things where you can see from things very fast on the picosecond timescale, bond vibrations, very fast motions, over up to real-time folding events or unfolding events. And you can probe them, and you can see this at unprecedented atomic resolution. So therefore, I think NMR, if you have a dynamic system or are interested in changes, is a method of choice if you can get stable samples. And this is what we actually did in this project in the next step. We kind of looked at the different timescales. And this is a classical approach, kind of so-called how flexible is the backbone, how dynamic it is. And on the left-hand side, we kind of measured the so-called head NOE, which gives us an information of the amplitude of motion on the pico to nanosecond timescale. So this means the green color scheme indicates, okay, there's a certain flexibility. And you can directly see, yeah, of course, not so surprising, in certain loops, in certain regions at the terminus, there is a high degree of flexibility because they are not stably folded. But we were also intrigued, we saw here in the middle where we expected actually a stable fold. some indications of some flexibility. And if you use other types of experiment which probe a slightly different time scale, in this case, more the micro to millisecond dynamics, we as a so-called R1 row experiment, we also saw, yeah, it's generally there are quite a bunch of motions in this protein when it's just in its free form. But they seem to also cluster here in kind of the corner where all these things are connected, many things. We then also wanted to know, is it just restricted to the protein backbone, or can we also learn something of the amino acid side chains? And for this, we specifically isotope labeled different methyl groups. And we also saw that while they were generally rather flat, they are not so highly dynamic, but in this corner, in this so-called sump region, because this DNA binding is described as a right hand, it has a palm, fingers, and a thumb. And in this case of DNB, it has an additional associated domain, which is shown down here. It was clear that it's needed for DNA binding, but its exact function remained a little bit obscure. So in the next step, of course, the first thing we wanted to do is to test kind of DNA binding. There were crystal structures with DNA, and we simply used this template and just looked, okay, how does it affect our DNB in solution? And as we could see, we could map, and this is shown here at the bottom with the green gradient, we see clearly chemical shift changes, and they map actually on the whole protein, which I described as the hand, but also in the associated domain. And you see the chemical shift changes, mostly kind of intensity changes, etc. And also we clearly see, if we kind of chop off the C-terminal associated domain, we still see binding, but nevertheless, even though we see a strong binding in the full-length protein, this associated domain on its own cannot bind the DNA. So our working hypothesis here was at this point, okay, yeah, well, this kind of C-terminal domain is kind of needed to lock things in. It cannot bind its own. It kind of, once the DNA is bound by the protein, it locks things in. And also, if we kind of checked computationally kind of the charge distribution, we clearly see you have a positively charged groove where obviously the DNA with a negatively charged phosphate backbone can very nicely bind in, and it's kind of matched very well. So we thought, great. And what about dynamics? Because of course we had this highly dynamic protein which experienced motions on several time scales, but what happens once it binds DNA? To cut a long story short, and we did the same analysis like before, we clearly see, as you see here on the bottom, we kind of lose a lot of motion. It rigidifies, it kind of locks in to a certain extent around the DNA, which was at this point also not surprising. But one thing which had surprised us, we would have expected, okay, it might be like a rock. It might be then once it bounds the DNA rather stable and might be only certain flexibility for its enzymatic function. But we were a bit surprised at this point that you can appreciate that we still have certain dynamics on the surface of the protein, on the outside, opposite of the DNA binding. And we were a bit surprised at this, and we weren't sure what might be the reason or what might be a functional implication about this. I will come back to this. And as you can see here, also we have a very strong binding. We use biolayer interferometry, where we can kind of, with these sensors, tip in the solution and measure the on and off rates, similar like to SPR, and kind of get kind of all these kinetic parameters. And we clearly see we are in the low nanomolar range, which also was expected because we see a lot of peaks disappearance in the NMR titration. So we thought, well, cool, we have an understanding what comes out. So now I have to tell you a little bit about the limitations of NMR. As you can see here, these are 3 proteins from our lab. It's at the moment not important what they are, but you see, and so far I've presented mainly spectra of the protein backbone, the so-called 15N spectra. They have a problem. The larger you get, you have run into 2 problems. You run into one problem is at one point you get too many peaks and they might overlap. with some tricks, adding D2O to your medium and deuterate the side chains. You can alleviate this because it narrows the peaks, but at some point your particle gets too large. For example, to 1 hexameric protein we have in the lab at almost 300 kilodalton, where we can see, if we are very positive, 20 resonances, which we cannot really assign. We don't see large parts of the protein. But luckily, there are ways around this, and these were kind of pioneered end of the 90s in Louis K labs in Toronto, and several other groups have kind of contributed to this. You can actually, in a highly deuterated background, specifically isotope label the methyl groups. And they have two key advantages. You have 6 methyl-bearing amino acids. The methyl groups has 3 protons. It gives you, in general, in theory, 3 times the signal, and they're normally rather free rotating. So they give a very strong signal. So in Louis' lab, they have shown they can even get good spectra of this kind for kind of the proteasome with associated particles up to 1.2 megadalton. So super huge proteins from the NMR perspective. And this is also great because they are also come in certain regions, as it's depicted here out of a review out of Jerome Bouboyer's group. They come in certain groups like the methionine methyls, the isoleucine, threonine, etc. So you get some information. And just for reference, I show the largest protein we're at the moment working on is the human lawn protease at 600 kilodalton. And this is the isoleucine spectrum we measured the other week. So you get very beautiful spectra, even for large complexes. So which gives us kind of a handle to study these large complexes as well. So, and therefore we also went to the assignment for the DIMB of the methyl groups. So with tedious, we kind of combined it with kind of the standard toxi assignment theory and via approach and via methyl-methyl NOEs. And I think we ended up to almost 95 to 98% assignment. You see the assignment here, they are nicely distributed all over the protein, just color-coded as on the outside. And then we looked, actually, what do we see? Where do we see interactions? And this came a little bit as a surprise. We rather expected, okay, these PID domains, this PAD domain might be a bit separate, but the other one looks like a very compact core. But we were a bit surprised that we see all of these gray lines here represent interatomic distances that we could detect. And you clearly see that in the individual domains, you have several of these interactions that are stabilized between the secondary structure element, but in between the elements, there is only here a little bit to this so-called sum domain. And this sum domain was the one which had this excessive dynamics. And so we wondered, okay, might this somehow be related? Because we rather expected a more complete picture, that there are more crosslinks. And here it's kind of shown these interactions. Hopefully you can see. So, and in the next step, we wanted to know, there were some older papers indicating that DINB might be interacting with RNA polymerase. The E. coli RNA polymerase is composed of several subunits and about 300 kilodalton in size, so a large protein. So we initially just used different DINB constructs and checked how they are interacting with this. And what we mainly saw is that already at very low equivalence, we lost all signal of the backbone. We had very tiny titration steps to see something. So we just could confirm, yes, it binds, and it binds rather excessive and strongly. And we could only see from this that the PID domain, the PAD domain might not be involved. So interesting, but we wanted to see, can we squeeze more out of it, get more on this? And there comes the methyl groups I just talked about into play, because they're more sensitive, and we used them for the same experiment. And here we could clearly see that it was rather specific, kind of this core domain out of the right hand with the palm fingers and the thumb were involved. And if we kind of looked at the strongest effect, we could see on the outside actually a patch which kind of interact with RNA polymerase. So, and when we check back, this was actually this pad where I said upon DNA binding, it shows still this enhanced dynamics. So we propose that these kind of effects might be additive, that there might be a connection and a communication with these, because DNA binding as well as RNA polymerase binding doesn't exclude each other. And there are also, the RNA polymerase binding is also in the low nanomolar range. To sum up a long story short, of course, this is a cartoon. The model we have at the moment is that kind of the DNA binding is facilitated by the inherent dynamics, and it's kind of locked in by the PAD domain. And this process can be kind of sped up or made stronger in the presence of RNA polymerase because the motions in the kind of backbone of this right hand are reduced. So we are also working on several other proteins. They are even larger than our DNB, which is shown here on the right-hand side. We are working at the moment on NAS-A, 55 kilodalton, and UVRD 70 or 80 kilodalton. So, but we are not so far that we have a full story. So I will switch gears slightly to the other interests in my team. So we want to understand how protein quality control works. As Vicky already mentioned, I worked quite a bit before with molecular chaperones. And kind of for me, it was a natural step to also look at proteases. And we at one point decided to work with a mitochondrial human HtrA2 protease for two reasons. A, mitochondrial proteins are still very well expressed in E. coli, make it they're amenable for isotope labeling. And for several other questions, this was an interesting kind of target for us. So what role does HtrA2 play? Well, it sits normally on the inner membrane in the mitochondria, is membrane attached, and does certain normal protein quality control jobs, kind of getting rid of aggregated protein. But upon apoptotic stress, it's kind of cleaved off, it's kind of secreted to the cytosol, and there it's an important kind of regulator or activator of the apoptotic cascade, because it kind of cleaves inhibitor apoptosis proteins, which then release caspases, which triggers a whole process. Even though on this textbook knowledge, this is quite well understood, but how this protein works was for a large extent very elusive. In last year's Lewis K group worked quite a bit on this protein as well, and together with us, I think we have now a very good understanding how this works. So, and this is the work from other student in the lab, together with Jens, who was on the DNB project again, Emily, and it was her PhD work, and I think she did a tremendous job getting this complicated system working. So initially, we kind of also exploited in a similar manner the modular architecture of the protein, because this protein has the protease domain with a certain regulatory loops and the catalytic triad, depicted here in the bluish color and shown down here, as well as the substrate recognition, so-called PDZ domain. And we said in the beginning, okay, we focus on the PDZ, how things are regulated here and might be transmitted then further on to the full-length protein. So we did what you do as an NMR spectroscopist. You first have to see if you get good spectra, and then you have to assign them. And they matched quite well, actually, with the crystal structure depicted here again with the C alpha C beta shift. We only saw that here this beta 14 strand is not really formed in solution. Looking at the crystal structure as shown here gives us some hints, because this kind of beta 14 sheet is kind of packed on the protease domain and is supposed to be kind of changed upon activation where the protein is supposed to open up. So it made some sense that this might, in this isolated form, not stably formed. So we also used our toolbox of the dynamics measurements. We initially kind of study it. We see in general our protein is very rigid, very stable, not a lot of pico to nanosecond motions, as depicted here of our head NOE values. Here is a flexible part which is not stably formed. It's a bit lower. This makes a lot of sense. But if we looked a little bit on the micro to millisecond dynamics, we saw that we had here a longer loop and a little bit this helix 5, which seemed to have some interesting behavior. And we kind of probed this a little bit with different techniques. And we got kind of a little bit that there are certain dynamics in this loop adjacent this helix 5. And we also observed that this helix is not really stabilized by other inter-atom contacts, the NOE contacts. So we postulated or hypothesized at that point, okay, this might be maybe something important in this helix 5, and I will come back in a bit to it. So it was also known from biochemical assays that you can bind activator peptides to this PDZ, which then trigger activation of the whole protease and the full-length protein. And we wanted to know actually where do they bind? Is there certain things associated with this? And we kind of both checked the protein backbone as well as the methyl side chains again to map this interaction. And it kind of is a certain groove within this PDZ, which also then is postulated to trigger an opening within the full-length protein. So in the next step, we also looked at the side chain dynamics, having this very nice methyl kind of labeling, which also revealed that most of the protein is rather rigid. It's stably formed. There is a certain dynamics here at the termini and the surface, which also is explainable. But also, if you looked again on the methods, we also saw this micro to millisecond dynamics, actually, which was very confirming for us that we also observed the same likely the same process on the side chain that I just talked about on the protein backbone. So we thought, okay, there might be something with this helix 5 and the adjacent loop. But at that point, we didn't have a really idea what this might be. So there were some literature floating around which postulated some of these class of proteins, these high temperature requirement HtrA proteins, might be modulated by divalent ions. And at one point we said, okay, well, we anyhow need to run some activity assays with a full-length protein. Maybe we simply test if there's an effect of divalent ions. We just simply choose the four most common ones: magnesium, calcium, copper, and zinc. And I have to add here, these protease works in the absence of ATP. So you don't need to worry about magnesium, which is already in your buffer based on the ATP. And when we run just a gel digest, which you see here on top, on beta-casein, we clearly see, yes, our protease cleaves. You see the cleavage product showing up already after 10 minutes. And you have an effect, and it seemed to be a bit faster in the presence of calcium and magnesium on the one hand, but on the other hand, if you added copper and zinc, you actually abolish the function of the protease completely. So we said, okay, cool, we can modulate this thing. Let's dive into it. What might be kind of the cause for this? And if you quantify this, you see it's slightly enhanced under the conditions we used for calcium compared to kind of the addition of no ions. But on the other hand, you have a almost complete abolition on copper. So we ran a coupled assay using an activator peptide and a self-quenching fluorescence peptide, which is depicted in the cartoon here. And we ran it in different conditions. We ran it also on the one hand, varying our cleavage, kind of cleavage or substrate, which gives us some indications on how the effects. So there we see maybe a kind of a 3- to 4-fold increase of the cleavage rate of the catalytic efficiency. But we wanted to know, okay, we can modulate it with calcium and magnesium, but what is the reason? Because you can imagine many different things this might this might alter your proteolytic cleavage. So at this end, we thought, let's see if we can also, with varying the activator peptide concentration, get some insight, because with this we can indirectly extract the KD. And this turned out that this is likely the case, because we also got here a way larger effect, because we went with subsaturating amounts of our activating peptide, and we actually reduced the apparent KD from about 25 micromolar of our activating peptide to about 2 micromolar, a factor of 10-fold. So we know, okay, and one other interesting finding what we could with this assay, which I would like to add, is that we could also reveal why copper is inhibiting. It actually, we could with high activator peptide concentrations displace the copper and relieves this inhibition effect. So we got kind of a rather good picture on what these things are happening. But of course, we wanted also to understand it in a more structured dynamical level on the full-length protein. But first, we kind of looked at where the calcium might be binding. So we titrated initially the PDZ domain with calcium and all the other ions, and we saw very distinct effect here in our kind of close to helix 5 in this kind of loop region, where we also saw the enhanced dynamics. As it was a little bit spread all over and on the messages was not so clear, we also confirmed this in a full-length protein using gadolinium, which has a stronger effect, and you can really nicely pinpoint, as the late Ivano Bertini has shown many, many years ago in Florence. You can use it to kind of map rather exactly calcium binding sites. This was the word I was looking for, I'm sorry. So with this, we got an idea. Okay, it's binding to this 5 and might trigger things, and this also brought us over to the full-length protein. So we started looking at the full-length protein. So here in blue, you see the full-length mass spectrum of the full-length trimeric HtrA2. And in yellow or orange yellow, you see upon titrating an activator peptide. You can directly see, yeah, not everything changes, but you have significant changes, which is annotated here. And if you looked a little bit closer at the different things, we kind of see different effects. On the one hand, we see here at the interface between the green PDZ substrate recognition and the blue protease domain, this is only a single subunit of the trimer for clarity. We clearly see a lot of signals disappearing and shifting, and also with work from Lewis K lab, it's clear this interaction is broken. It separates, it opens up kind of the entry to the protease side. But we also see, and it's known for these HtrA proteases, they have several regulatory loops which have to kind of rearrange to form the active protease. And we clearly see also in these a lot of things happening, which we kind of track. So here we have only changes, chemical shift changes, but we also want to get a little bit of structural information. And for this in NMR, you can measure distances between two atoms, and these are called NOE distances. And we also check between methyl groups the NOE distances. And this is shown here. And we clearly see within, not just within the breaking of the two domains, but also within other parts of the protein rearrangements, depicted by a cluster here close to these loops, where you see in blue the apostate, and you see quite differently several of these are gone. Of these NOEs, kind of that there are changes and there might be more flexibility in the activated state. Of course, it's very difficult. If you see new peaks, you can assign it always more easily than if you're just disappearing peaks. It could always be that your sensitivity is not strong enough or other issues happen. Therefore, you need an internal control. And luckily, at other sites, like depicted here on the right-hand side, we more or less see exactly the same NOE patterns in the APO and the activator peptide bound state, kind of giving us some support that what we are seeing is not just effects on base that we simply don't observe, it's we lose sensitivity. So this kind of gives us, besides the chemical shift changes, some indications about the changes ongoing. And this is kind of a summary slide of this. It's kind of the interactions we observe, like before with the gray lines. You have kind of these two effects. You have this in purple, seeing the rearrangement of the loop and kind of the breaking of kind of interactions, and a rearrangement of things we observe in yellow based on kind of the addition of the peptide, which kind of enabled us to propose a model. What we think is happening initially binds the activator in the groove between the two domains, leading to the opening. You have a domain separation. This kind of triggers because it frees this so-called L2 loop and triggers a kind of a cascade that these loops can rearrange and kind of form the active site. And to our surprise, we also observed something at the far end terminus, that we lose some interaction between this helix 1, which was a little bit unexpected, that we have kind of effects through the whole protein. And we wanted to really look what might be the reason for these effects on this helix. And for this, we used something called paramagnetic relaxation enhancement. In a sense, it's based on the fact that you have an electron spin in this radical, this MTSL molecule, which kinds of locally leads to very fast relaxation of your spins you observe in the NMR. And this MTSL is very simple. You can couple it to cysteine. And so we made single cysteine mutants of our HtrA at the end terminus, adding a cysteine in there and just looking at the effects. So just focusing here for simplicity on the isoleucine region of the spectrum. Well, if I have the apostate, I just see for certain things kind of a relaxation enhancement, which in the end you simply see that your peak is broadened beyond detection. Like in this case for isoleucine 150, which is very close to this S145 and also to adjacent. So we wanted to know what actually happens when we add calcium. Well, we see a little bit stronger effect, a couple of residues. But what actually was very telling and surprising is upon addition of the activating peptide, we saw a significantly larger effect. So meaning that the flexibility of this N-terminus is greatly enhanced. So of course we quantified this and kind of put this on a structure, and I will guide you through. So here on the left-hand side, we have the apostate, and we have only a local flexibility. Possibly only this first part bearing the empty cell can flap around in the trimer. Whereas on calcium, there seems to be clearly that partially opening of the cleft between the PDZ and our protease domain is already occurring. So indicating this might be the reason why we get an enhancement of peptide binding, because it's already partially opened. And upon activated peptide, we see kind of effects all over the place in the interface, suggesting that this helix is rather flexible and can enter also in between. So in summary, cutting a very long story short, we believe that we have now understood a little bit better how this HtrA2 is regulated by the different things. And we also think that certain aspects are promoting and hindering this activity. On the one hand, you have calcium, enhanced calcium, which is also countering if your mitochondria breakdown, enhances the cleavage rate and kind of promotes apoptosis. On the other hand, if you have zinc, and this was shown mainly by Lewis, case group that there's an inactive hexameric state, which we have also some data indicating that this promoted by the presence of zinc might inhibit our HtrA protease. So just for the final minutes, and I hope I'm not over time yet, I want to give you also some flavor of some things we tried together with the group of Paul Chanda in Vienna. Because, well, as you saw, the problem for these large complexes is you see at maximum the meso groups, but you don't get so much information if you don't take the separated domain of the protein backbone. And we had a kind of a system we worked on. It's a so-called peroxyredoxine. It's important for removing reactive oxygen species. They have kind of two cysteines, which can be then kind of oxidized and reduced and need to resolved by a cyredoxin system. And there are kind of important aspects and also in protein quality control and aging. But these proteins are kind of built up of dimers in the most simple state, make a decameric state for the yeast protein we worked on, and can even make larger complexes. And we struggled for some time to get really good solution data, because they can form these different large structures. And it's very interesting because there are also connected. Some indications say they might have some chaperone activity under certain conditions. And this was actually our starting point. And this out of a review a former postdoc Laura wrote a couple of years back. And well, but really what is going on besides structures was not really known. And we wanted to contribute to this. And at that point, we teamed up with Paul Chander, and just a very brief introduction of solid state NMR, which we did with Paul together. Well, on the one hand, you need less and less material nowadays. This is a little bit outdated. You have these small rotors. This is still a 1.3, which I think we used for the study, but nowadays they're getting even smaller, 0.7 or 0.4 millimeters. You need only a very few microliters of sample, and you have a high spinning rate, and you get very, very narrow lines. If you have a homogeneous good sample, you have about 50% of water. You sediment with ultracentrifugation, you have proteins in there. And you can study. We did parts of the studies we do here in Sweden, up in the north in Umeå, where they have good equipment. But for this study, we went to kind of Grenoble and Vienna, working together with Paul. And we wanted to study the different states of this protein. Because as I said, it can be reduced, it can be oxidized. There are crystal structures available, but they indicated that the oxidized state is highly dynamic, flexible, but nobody really understood what might be the reason. So we said, okay, this might be a nice project to do solid state NMR, and actually turned out a very beautiful project for us. So shown here, and the work was mainly done by Laura, in the reduced and the oxidized state, we got rather decently spectrum, very nice one in the reduced, you'll see nice lines. And in the oxidized, you see some broader lines indicating some heterogeneity and maybe also dynamics. At that point, we didn't know. We tried to assign these. We got about 75% for the reduced state and about 50% of the in the decameric state of the oxidized state. And then we looked actually what part in the oxidized state we don't see. Is it just random or does it cluster? And actually it turned out we actually see on the outside close to these cysteines, which we could not assign in the oxidized state where the disulfide is formed. This is the important part. And well, being interested in dynamics, we kind of used a lot of different dynamics to probe what might be the effect. We probed different time scales. And it turned out that the nanosecond dynamics were rather similar. There were some small modulations in both stages. The color coding for yellow and red stays the same for this part. But we clearly see in the microsecond dynamics, huge microsecond dynamics enhancement in the oxidized state all over the protein. Of course, we don't see it here at the C-terminus because we lack the assignment. So, and we wondered, okay, can we quantify this? Can we really get a handle on this? And actually, Paul used a so-called detectors approach, which is a more sophisticated approach. You normally analyze dynamics data and kind of assume a single process. Here, we kind of try to decouple it in different processes or different timescales. And we see kind of effects here ranging from the very fast nanosecond timescale also in the microsecond, but really a kind of different microsecond timescales and processes ongoing. And we wondered, okay, what could be the reason for this? And we also looked at this and looked with other probes, and we also saw another process ongoing, which we kind of used with a slightly different approach, which indicated that we have at the dimer-dimer interface some motions. And this kind of gave us a hint, okay, maybe if we can break down the decamer, we can also get some insight into the inherent dynamics within the dimer. And there, well, our local solution NMR expertise came in at the end, because we also realized that we could break down the decamer if we kind of heated up the sample. We measured diffusion, which gives us a hint about the size of the particle. Depicted here in the solid line would be just based on the change in viscosity. If you heat things up to 60, would be kind of the curve you would expect of the diffusion coefficient. But actually, we see a transition. And at 60, we actually are in the dimeric state. for our TSA protein. And so we thought, well, maybe we can study this TSA in solution at high temperature. Well, in the end, it turned out the wild type was not so stable, but we could make a single point mutant where we changed an serine to an aspartic acid. And if you look at the mass spectra, they overlap very well. Here in blue, you have our mutant, and here in red, at the different temperatures, our wild type. And you clearly see the large particle is a little bit heterogeneous in solution, but it becomes the dimer and with nicely overlapping. So we could also use, in this case, our methyl groups to get some insight. And we measured kind of the side chain order parameters, which give us some insight about the very fast motions, pico to nanosecond timescale, and correlates this to what we have seen kind of on the protein backbone. And we already saw that we have here towards the C-terminus a very flexibility in the oxidized state, which we were able to study in kind of solution. So, and we wondered, what might be the cause? What might be the reason, what triggering? Because normally you would expect a disulfide is formed, it certainly stabilizes the region. But it turned out, if we ran the analysis about protein frustration, kind of pioneered by Peter Volines over the years, and we realized that actually in this region, when the disulfide formed, so-called local frustration is quite large, quite enhanced. What is local frustration? You mean you bring amino acids together, charged or hydrophobic, which doesn't really match together. So, for example, in a large charged patch or hydrophobic part, you kind of bring in a hydrophilic amino acid, and this creates these kind of frustrations that they don't want to be together. And with the disulfide, you form this. So, in a sense, what we see in the story, and where kind of it was a beautiful combination of solid and solution state, is that forming the disulfide kind of brings the strain to the protein, and which kind of manifests enhanced microsecond dynamics. Well, actually, it turned out in the end, we were not the first ones to observe this in this TSA. I think there was a paper a couple of years or 2 years before on a sulfur reductase, if I'm not mistaken, they kind of have seen similar things. So this might be a common topic or a common theme that disulfides might be, for certain reactions or certain proteins and functionalities, actually creating a larger extent of local dynamics. And this might be related to functional repertoire. And well, of course, this is no structure. This is a cartoon and an illustration only. So with this, I hope I have given you some thoughts, some things that NMR, besides the beautiful approaches and beautiful kind of steps Cryo has taken lately in structural biology field, NMR has a big role to play, in particular revealing the tiny little dynamic details, which actually are, I think, in many cases, really, really important for biological function. And with this, I would like to thank my team, depicted here on the left-hand side, and also the former team members who did lots of this work. Our collaborators, I mentioned Paul Chander in Vienna, but also our other collaborators, Edo Pinjakuda, for which we work on an other, and Anja Böckmann, different other solid state projects, some local things, studying also chaperone things, and most importantly, parts of the guys next door at the Swedish NMR Center, which are always very helpful and give us sufficient measurement time that we can measure our stuff. And with this, I would like to thank you for your attention, and I'm happy to take any questions.
So thank you, Björn, for a fabulous talk. What a tour de force across lots of different NMR techniques. We've now got time to welcome some questions for Björn. So do just type it into the question box as shown on the image on the screen. We've already actually had lots of questions come in during your talk, so we can get started straight away. I'll start with a general one. Someone writes, can methyl NMR be combined with other biophysical techniques to provide a more comprehensive picture of protein dynamics?
Sure.
I mean, have you worked with particular other techniques and done combinations that you've seen work well?
Yes, of course. In general, you can combine it with most things. Not presented now. We also do a little bit of SACs and doing some native mass spectrometry for larger complexes, kind of getting an overall picture. And of course, you can also combine it to structural studies with X-ray and cryo-EM. It always depends on your question.
Okay, then we've got a couple of people asking about the divalent ions. So one person asks, given the importance of iron in mitochondrial function, for example, heme synthesis, did you measure the effect of iron 2+ on HtrA2 protease activity?
No, iron we didn't measure. Actually, I don't know why. We simply started with these 4 I presented. And this was kind of initially started as just out of curiosity, and then we could kind of manifest some effects in it. But of course, it's a very good point. We might need to look at iron as well.
Okay. And then another question on that topic, can the proposed functional role of divalent cations be used to cure mitochondrial defects such as Friedreich ataxia?
Well, I will answer rather generally, possibly. Yeah, it is. It seems to be, and this is also very preliminary, this is not a common thing that all or other HtrA proteases are activated by similar mechanism. I think it's reported that I think some bacterial ones are also inhibited by zinc and copper. This we also see, but this activation so far, at least for the ones we tested, we have only seen for HtrA2. It might be unique, and it might then, of course, also be exploited.
But I guess I could imagine in a therapeutic way, it would be hard to target potentially.
Probably, because I could imagine that altering kind of iron levels might be A, very difficult, and B, might also create lots of effects you might not want to.
Yeah. So then we have another question heading more in the cancer direction. So would peroxyredoxins, can they be used for early detection of DNA damage and therefore identify those at high risk of developing cancer?
Very good question. Yeah, well, it's a rather C-reactive oxygen species, rather small molecule. I'm not so sure if you could exploit them, but of course, maybe spin it a little bit further, kind of exploiting things where we haven't really touched it. And I think this is also done, this DNA repair mechanism, this kind of polymerases or kind of detecting mechanism of DNA damage might be rather, in my view, and this might be also limited knowledge of the whole system at the moment, might be exploited in a better way to kind of be detectors for DNA damage and therefore also early detection.
Okay, then we've got a nice question here, possibly from a student, clearly interested in what you've been showing. What kind of skills would you recommend a third-year biochemist undergraduate develop in order to enter this field of study?
Well, I think the most important skill is a good hand on the biochemistry. So because well, this is only possible. You need stable and good samples, and you have to produce several of these, several labeling schemes. And if you are good in the biochemistry, I think this is what I call half of the way. It's always good, of course, if you have a genuine interest in this biophysics and understanding, but well, unfortunately, we cannot just take the samples out of the fridge and have them there. We have to produce them, and this there you need persistence, and this is actually, I think, the most important part, in my view.
Okay. And then we've got someone going back to the PAR-B. What is the role of PAR-B proteins as part of a cellular machinery involved in molecular genetics in the discovery of drug inhibitors against cancer? It's quite a...
PAR-B. Okay, I think it's a DIN-B.
Sorry, PAR-B.
Okay, so yes, these their role is they kind of detect, or together with RNA polymerase, this is a bacterial system I worked on, but there are slightly related human systems as well. They detect DNA damage and they repair it. They are not super good in the way that they have a super proofreading. They simply repair the things. They are also kind of coupled to evolution because they miss certain things, but of course, you can design them and better understand to kind of detect these damages and, for example, give you a readout. This is definitely, I think, possible, and you might also engineer the system. But as I said, even in bacteria, you have not only likely the three systems out of the review paper I just had as my intro slide. They might be interconnected, and I think the detailed understanding what comes when, this is at the moment, even in the more simple bacterial system, not 100% clear.
Thank you. I'm just going to finish off with another sort of NMR question, really. You mentioned in-cell NMR at the beginning, and I'm just curious, kind of, firstly, what sort of system have you been doing? Have you been doing the sort of overexpression style of methods, or the injecting into oocytes? And sort of what kind of things are you finding? Are you looking at the same proteins and finding that they behave differently in the cell, or have you been using these on different projects?
Yeah, well, we use it in slightly different projects. Well, for in-cell NMR, we normally do the injection method. We so far used only kind of what was kind of introduced or pioneered by François Tellet and Phil Zelenko's group, kind of the injection into HEK cells and then studying whatever protein we put in or interested in. So this limits us a little bit to kind of small proteins, IDPs, and then you can couple it to cell biology and kind of understand what kind of might be the interacting partners and how they might modulate this. Actually, we used it in a study on alpha-synuclein a couple of years back. So we are not really active on the in-cell part. We might take it up now we got a new student in the lab and some questions. But what we are actually at the moment trying to do, and we have a little bit on the bacterial system, we use kind of small vesicles, which have compartments of cells or cellular compartments as kind of nanotools or whatever you would like to call it, to kind of simplify the systems, because we realized if you use a whole cell, it might complicate things, and you might not get sufficient resolution to get the information you might be interested in. But there will be a little bit more coming, hopefully soon.
Okay, well, thank you very much again, Björn, for your talk and for answering all these questions. I think we've all learned a lot and enjoyed hearing. Thank you also for all of you who've attended, and if you want to continue the conversation online, then you can do that by following @BiochemSoc and @PP Publishing on Twitter or X. We do welcome suggestions, as I mentioned earlier, for topics and speakers for future webinars, and if you have an idea for 2024, then we do invite you to submit a proposal for an upcoming webinar. You can find more information about the webinars and propose your own one, or watch previous recordings at www.biochemistry.org. And the recording from today's webinar will also be available to watch within the next couple of weeks. Do join us again on the 1st of May at 2 o'clock British Summer Time for our next webinar, The Complex World of RNA Modification and Replication. We will have 2 early career researchers investigating how the ADP ribose cap functions in human cells, as well as advancing our understanding of penovirus biology. For anyone who hasn't yet heard, the registration is now open for our brand new Biochemical Society Festival, which will be taking place in London, and we will have an exciting 3-day meeting celebrating the vibrant spectrum of molecular bioscience disciplines, as well as interactive workshops and networking events. So more details on the screen and obviously on our website. Finally, I just want to highlight that it's more important than ever that we all stay connected and engaged with fellow molecular bioscientists, to join the Biochemical Society's community of researchers and specialists and take advantage of key benefits, which include discounted registration to society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of our journals, and more. So do go to our website and find out more about all of the benefits that you can have by joining the Society. Thank you to everyone again, and goodbye.